Welcome to Brain Ponderings. I'm Mark Matson, the host. This will be the first in a five-part lecture series on brain aging. The first lecture will focus on general theories of aging, how gerontologists and people who study aging uh, try to understand what is happening during aging in organ systems, cells, the molecular level, and how they go about this. And then it'll also include animal models from there. The next lectures will focus on cellular and molecular mechanisms, on the functional and structural changes that occur in the brain during aging, as well as age-related neurodegenerative disorders. And the last lecture will be on anti-aging approaches such as exercise, energy restriction, using your neural networks, how those strategies can counteract aging. I'll also talk a little bit about other approaches, including dietary amino acid restriction and perhaps some specific chemicals obtained in the diet or produced by bacteria in the gut. So the first lecture I'll get started on here. This is a drawings, paintings actually, by William Udermolen. He was an American artist who did a lot of portraits, and these are self-portraits, um, beginning before he had dementia, and then at different stages of dementia. He was actually diagnosed in 1995, but he had memory problems for at least 4 or 5 years before then that were noticeable. And you can see the dramatic deterioration of his ability to draw, something he'd done all his adult life. Right. So one wonders then, uh, why is it that this can happen to brain and body as well during aging, the decline, which is progressive, unavoidable, at least thus far, and probably for quite a while into the future. Well, I trained as a developmental neurobiologist, trying to understand how nerve cells are born, differentiate, form connections, integrate into circuits, and then function during life, and then either do or don't degenerate late in life. And one thing that's noticeable when you study the formation of the brain is it's very dramatic, highly uh, orchestrated, and it occurs very quickly. We did a lot of work on rats and mice, which in 20 days or so, they go from that fertilized egg to newborn. And during that time, millions and millions of neurons are generated from stem cells, and connections form and become functional. And then one wonders, why is it that this structure and functionality of the brain, which arises so quickly and precisely in many ways during development, cannot maintain itself throughout adult life. So this first lecture is going to focus on theories of aging, mainly from an evolutionary perspective. And then the second half of the lecture will be on animal models of aging, from worms and flies to rodents, non-human primates, and humans, which are, of course, it's harder to study aging when you really want to try to understand what's happening in the brain. And this is a quote from Francois Jacob, a Nobel Prize winner, that just what I said, it's amazing that this structure of the brain can form in such a precise manner during development, but it's not able to maintain itself, and we all age and die. So most of you know this, that age-related neurodegenerative disorders, Alzheimer's, Parkinson's disease, and then also stroke, which is a major cause of disability and death throughout the world. These three brain disorders of aging account for nearly, it's getting close to not half yet, but maybe 40% of all deaths. And as people are living longer because of advances in cardiovascular disease, diagnosis, treatment, as well as cancers, uh, those advances have led to actually more people getting Alzheimer's, Parkinson's, and stroke because they live long enough to get into the age danger zone, if you will, of 70 and above for these disorders. And there's no treatments yet that have any meaningful impact on the progression of these disorders. And early diagnosis, there is advances, but that's still a problem, and we don't have any treatments. But as I'll talk about, particularly in the last lecture, there are ways to reduce risk and counteract some of the processes of aging that seem to be fundamental to these disorders. Okay, so how do you define aging? One kind of simple definition is it's progressive molecular and cellular changes that lead to the death of organisms. With this definition in mind, successful aging is aging that occurs without major functional problems. In the case of the brain, it's able to control your body movements, receive signals, and process at them and have outputs. So things like learning and memory, regulation of mood, and essentially all behaviors. Um, so one of the goals of aging research is to enhance health span, the functional uh, lifespan, if you will, of an individual. And I'll talk about in a little bit average lifespan and maximum lifespan. So in the case of humans, average lifespan is typically around 80 years, maybe a few years less, depending on what country and the conditions people grow up in and experience during their life. And the maximum lifespan, so far, it's 122 years, the longest lived human on record. Okay, so unsuccessful aging is unfortunately common. It's when diseases set in, often in mid to, I guess you'd say, early old age, 50s, 60s, and then certainly 70s, developing chronic diseases, whether it's cardiovascular, um, inflammatory diseases, all sorts of things, liver, kidney, and cancers. Okay, and then from an evolutionary perspective, one considers differences between uh, aging of the individual and the species. Um, Individuals age and die, whereas in some ways you could consider a species potentially immortal, although uh, historically most species become extinct and are kind of superseded by other species that may have have evolved better survival and reproduction abilities under the environmental conditions where they exist. Um, are there immortal cells? Well, I guess one can consider oocytes immortal in a sense, uh, passing on the DNA from generation to degeneration. Stem cells, um, not really. Stem cells tend to burn out with aging too. That is their proliferative potential. Are there species that don't age at all? Um, Not really. One could make that case for certain plants, particularly trees that propagate, or other plants that propagate through rhizomes, sending out shoots under the ground. So, for example, the aspen trees, when you look at what most people think of as a bunch of aspen trees in the background of the green pine trees, say in Colorado, those aren't actually individual trees, technically. They're all the same uh, organism that are all connected by underground connections. Simple organisms like yeast, maybe bacteria, they undergo asymmetric cell divisions, and so when they're dividing right before, they segregate all the damaged parts, proteins, DNA, membranes to one daughter cell, which is usually the smaller one when the cell divides, and then the undamaged components of the original cell are maintained in uh, the larger daughter cell, and that daughter cell then goes on to divide, whereas the daughter cell, smaller cell with all the damaged stuff dies. And then there are a lot of species that we really don't know how long they could live uh, because it's really hard to study, certainly in the wild. But some of these seem to continue, fish and lobsters, they continue to grow throughout their lifespan, and they don't necessarily die by old age, but by um, other causes. Okay, theories of aging. I'll take a little bit of time to talk about these theories. Um, and so, what I want to do in talking about these theories is divide them up as follows, based on theories proposed by specific scientists throughout the years, actually, throughout the decades or even hundreds of years. Um, One old one is the program death and germplasm theory of August Weismann. Um, and this idea is kind of intuitively, it makes sense that uh, since it's important to preserve DNA and the species versus the individual, aging may be advantageous to that mechanism of evolution that we all know about because older individuals consume resources, food, and perhaps other resources in the environment. And this may be a disadvantage to the younger individuals because there's usually limited food supply and in particular area where animals live. So ideas here is pass on the genes that are advantageous for reproduction, which is kind of the key thing, reproduction and survival early in life to pass on genes. And this theory, kind of a sub part of this is that the cells of the individuals have a genetically determined lifespan and an ability to divide, and therefore they essentially, the aging is programmed. But this theory is now kind of disfavored, although it's something that gerontologists and people who study aging should keep in mind. Then another one, Medwar, had the theory of mutation accumulation that um, mutations occur in the DNA of cells, uh, and over time these mutations accumulate, and they're not selected against because early in life there's mechanisms that repair the mutations, or they just don't reach a critical threshold that leads to aging and death. Um, there's definitely evidence that mutations accumulate in DNA during aging, and a lot of those mutations are caused by free radicals, which I'll talk about a lot in the second lecture. Another theory proposed by Williams is that genes are advantageous early in life, and this is called antagonistic pleiotropy hypothesis. These genes that are advantageous early in life, for example, for competing for resources and mates and reproduction, those genes that have those functions become detrimental later in life. Okay, so that's an interesting idea, and there's evidence for that. And then finally, the disposable soma theory. Tom Kirkwood is kind of big proponent and developer of this hypothesis that germ cells are immortal and all the other cells in the body, brain, are disposable in the skin from an evolutionary perspective. So that, and this hypothesis, which again is not mutually exclusive with the other theories, damage to somatic cells accumulates throughout life, and mechanisms of protection and repair progressively fail. There's good evidence definitely for this, that the somatic cells function and structural and uh, progressively deteriorate during aging, whereas the germ cells, particularly egg cells, remain relatively unscathed. Um, yeah, and this, I just note Richard Dawkins' theory on the immortal selfish gene theory, and this fits in with the disposable soma theory. Okay, these are some of Kirkwood's reasons for rejecting the idea that aging is programmed, because it avoids overpopulation and eliminates post-reproductive individuals who would otherwise compete for resources with their younger kin, and the idea that it facilitates evolutionary adaptation by promoting succession of generation and fits the purported existence of aging genes. However, these these kinds of arguments are not really supported by the data to any meaningful extent. Um, so here's his arguments. If a genetic program existed solely to cause aging, any mutation that inactivated such a program would confer a selection advantage, and the genes responsible would become extinct. So that makes a lot of sense, that why wouldn't organisms just become extinct if there's a program for aging, because those genes would be selected for. Another argument against this is adaptive aging is based on the low abundance of visibly senescent animals in the wild, but animals in the wild typically don't die by aging per se. They're killed by predators, or they freeze to death or starve to death. And so that argument is not very strong. And then another argument is that there's uh, existence of an active set of genes control mechanisms that serve specifically cause aging and death. If these mechanisms existed, they would be susceptible to activation by mutation. And then finally, experiments show that if you if you mix long-lived uh, nematode mutants with shorter-lived wild-type nematodes, uh, the longer-lived strains are outcompeted in terms of reproduction and fitness. So let's just quickly go back through these. Antagonist pleiotropy, genes that are beneficial in early in life development and success in reproduction are detrimental to fitness later in life. And these would not be selected against, is the point here, because the detrimental effects don't show up until after reproductive age. And the disposable soma theory, um, again, here you have limited resources that are used to reproduction to pass the genes on, and that aging of somatic cells is selected against, sorry, is selected for in a way. So natural selection favors investment of limited resource into reproduction rather than somatic maintenance, accelerating damage accumulation and aging. So this is the molecular damage accumulation hypothesis for which there's tremendous support. And my second lecture will focus a lot on the hallmarks of aging, uh, particularly brain aging, that lead to accumulation of molecular damage at cell dysfunction, organism dysfunction, and what we see as the aging process and death associated with aging and diseases of aging. Um, and kind of a corollary of this, the regulated maintenance hypothesis, for which there's quite a bit of evidence that um, signaling pathways in cells, that is, pathways whereby they receive some signal from the environment. It could be a nutrient, an energy source, amino acids, other things. Um, these signals activate pathways and induce expression of genes or turn off certain genes that uh, that favor reproduction, and early in life favor somatic maintenance. So two pathways here are involved in cell growth that seem to actually accelerate aging are the insulin-like signaling pathway, at least in non-mammalian species, and the mTOR pathway. And I'll talk quite a bit about these later in today's lecture. So this slide is just going into this a little bit different and kind of merging two hypotheses, the antagonistic pleiotropy and um, the somatic cell disposability theory, if you will, the maintenance of and propagation of the germ cells across generations versus organisms. Uh, so there's some specific examples that early during life, calcium deposition in the building of bones is very important, but later in life, you can get calcium deposition in arteries and cardiovascular disease, for example. Um, and what I just said, that pathways that promote growth early in life may be detrimental later in life. Okay, and then finally, before I get on to a little bit more on general principles of aging and animal models, this slide points out that it's important to consider that um, most individuals die, at least humans, by acquiring certain diseases, whether it's cardiovascular disease, cancers, neurodegenerative disorder, kidney, liver disease. And these pathologies accumulate with age. And at some point, maybe the actual proximate cause of death, even though aging is the kind of initial cause or contributing factor to the development of these diseases. And in all of these disorders I mentioned, aging is the major risk factor. Although certainly you can get cancers early in life, um, and you can have mutations that lead to disease early in life, but those are fairly rare because they're selected against. Here's some examples of survival curves where you're just looking at the number of individuals that survive to a given age. So some species, for example, this curve here, insects, example where individual female may lay 1,000 eggs, and not many of them survive into adulthood, but those that do survive can can keep going. So, for example, um, tadpoles coming out of eggs and developing into frogs. A lot of those eggs that hatch and the tadpoles that come out of them are killed by predators early in life or die by other means, but those that develop into a frog live later. There are some species that also they live, almost all of them live to reproductive age and then they die right after reproduction. There's a few examples of that. Uh, this curve here is more like humans and mammals where you don't have much death early in life, and then at some point in midlife, deaths start to go increase, so fewer individuals are surviving. And then later in life, there's acceleration of number of individuals dying. Okay, so how do we understand aging? There's a number of different approaches. One is comparative biology approach. You compare the lifespan of two different species and then try to identify something that's different between the two species that might explain that. Um, Looking at individuals, animals or humans at different ages and uh, measuring something and correlating that with the aging process, and then ultimately seeing if you selectively manipulate that specific thing that you've identified as as being consistently associated with aging. For example, you can increase or decrease the expression of a particular gene that you think the protein encoded by that gene is a major factor in aging, and see if that manipulation of that gene affects the lifespan. And that's been done a lot, uh, more and more, particularly in nematodes, C. elegans, and fruit fly Drosophila, and mice. Can't do that in humans. Um, Search for environmental factors that affect lifespan. There are certainly environmental factors in humans that affect lifespan. Smoking, pollution, overeating can shorten longevity. And I guess you can make the case, and if you look at what's happening in the cells, indeed, we see changes consistent with accelerated aging. In humans, one approach that's applied a lot, uh, because it's one of the few approaches we can apply, uh, uh, is epidemiological approach, where you try to identify uh, differences in factors, environmental or genetic, that seem to be related to lifespan, Um, it's very difficult to do experiments to test whether indeed that specific factor associated with aging is causally involved in aging. And this is a big problem with epidemiology, that there are many, many studies that are published where the epidemiologists identify an association with factor X, say in aging, and then they don't show that that's actually any factor that's playing a role in aging. It could just be uh, happenstance that that's associated with aging and is not causally involved in aging. And then the intervention approach, which really gets at how do we understand what's really going on with aging, and are there ways to slow down aging, speed up aging, and then how can that be translated to humans? I'll talk about that in my last lecture. So comparative biology of aging, one can look at different species that have different lifespans and then look at a bunch of different traits. It could be morphological traits, behavioral traits, um, or organ function, or cell function, or gene expression, or whatever, and see if there's a correlation between a trait and lifespan between species. Um, Another is to take a species with exceptional lifespan and study them in comparison to, say, rats or mice, and see what differences there might be. Um, this slide shows that, and since this is neuroscience, mainly lecture, I'm going to focus a lot of the brain and not talk much about other organ systems. So there's some data where they make correlations between the brain size of different species of mammals and the gestational period, that's how long from fertilization of the egg to birth, the period of adolescence, and the period before the onset of puberty, um, and body size. And there are correlations if you look up across mammalian species with these four different features here: gestational period, adolescent period, onset of puberty, and body size. Um, and that's shown here, 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 and here. Nice correlations between brain size and these traits. So bigger brain is associated with longer developmental period, and often associated with that is longer period of a parent taking care, or parents taking care of and protecting the offspring after they're born. And then body size is generally related to lifespan, to brain size. Now that could be explained in part simply because it takes more brain to control the body functions, bigger motor or sensory cortex and areas that process that. But actually, a lot of that difference in brain size can be attributed to increase in size of structures having to do with cognition, learning, and memory, and complex processing of information. This looks at a number of different species. Of course, this lecture is on YouTube, so if you want to take the time to look at this in detail, uh, you can stop there and look at this. But this is just some examples where these kinds of data in the previous slide came from, where age at puberty, length of the growth period, uh, length of gestation are correlated, as I showed in the previous slide, with brain size, but also with lifespan in general. Um, there was some thought early on that, I'll talk about this in the next few slides, that there's a correlation between the metabolic rate of the animal and its lifespan. And that's true in some instances, but there there are there are many exceptions. For example, uh, birds, comparing birds to, say, rats, which birds can live a lot longer than rodents, even though they have a higher metabolic rate, higher body temperature. Uh, right, but that's actually an exception. I should point out too that individual cells, particularly mitotic cells, cells that divide, they seem to have a limited capability to divide. They can only divide a certain number of times before they stop dividing and die or become senescent. And interestingly, if you take, for example, fibroblasts from the skin of different animals that have different lifespans, the number of population doublings, that is, the number of cell divisions in culture, is related to the lifespan of the species. So if you take fibroblasts from human skin, put them in culture, if you take from a young human, they may undergo 90 population doublings in culture. If you take the fibroblasts from an older human being, say in their 70s, 80s, they may only undergo 30 or 40 population doublings. Okay, so that shows there's a correlation within the species, then between species. If you compare human to mouse, uh, if you take skin fibroblasts from a mouse when it's young, put them in culture, they only divide about 20 times. I mentioned this kind of exception of birds having a high body temperature and having relatively long lifespan. But for the most part, at least among mammals, there is a correlation where, for example, mice have a higher metabolic rate and they have a shorter lifespan compared to dogs or humans. Um, and interestingly, the lower body temperature among these mammals is associated with longer lifespan. And even in humans, for example, in the Baltimore longitudinal study of aging, in which individuals are followed from middle age until they die, individuals that have a lower body temperature, say 97.5 Fahrenheit, they tend to live longer than individuals with what would be often considered normal 98.6 body temperature. So there's that correlation. If you take invertebrate species, or even there's some evidence in rodents, and you reduce their body temperature throughout adult life, they live longer. And that's been shown in flies and worms and some in rodents. Also, one manipulation that extends lifespan in rodents, non-human primates, and probably humans, caloric restriction or intermittent fasting, that will lower body temperature. Um, if you have continuous daily calorie restriction, the body temperature is down uh, pretty much every day with, if you do, for example, in mice or rats, every other day fasting, then the body temperature is way down on the fasting day. And then it goes up and then back down on a feeding day. Uh, right. And in this study, they manipulated one of these pathways associated with aging, this insulin-like growth factor signaling pathway, which promotes cell growth, but seems to, in the long run, have a negative effect on aging, accelerate aging. And um, when this is genetically manipulated, um, then that you get a reduction in temperature, and that increases lifespan. Okay, now there's a couple really interesting uh, classes of animals that are kind of outliers when you look at some of these correlations between something, whether it's body temperature, metabolic rate, um, uh, and others on aging, and those are bats and birds. Bats have a body weight similar to mice, and then and yet some bat species can live 30 years. Uh, Actually, those may have body weight closer to rats, but mice and rats only live 3 to 4 years, and these bats can live up to 30 years, and that's the average lifespan. Similarly, birds, uh, again, which may have a body weight not so much different than a rat, say a crow, uh, they can live 30 years or more in the case of things like parrots or eagles, and yet the, the bats and birds have a metabolic rate and body temperatures higher than the mammalian species with similar body weights. So what explains these? We actually don't know yet, but one theory that developed had to do with this molecule, uric acid, which some of you may have heard of because levels of uric acid in the blood increase in individuals with gout. And uric acid is very insoluble in water or blood, and if it gets to certain concentration, it will precipitate out and form these crystals that can accumulate in, for example, areas of joints and cause uh, a lot of pain and inflammation. But it turns out bats and birds have very high levels of uric acid compared to mice and rats, a lot higher, like tenfold higher. And yet these animals live longer. So first that says that somehow they have a system for tolerating the uric acid. And uh, another is, uh, what is this uric acid doing when it's such a high concentration? Now this little chemistry or biochemistry here, UA stands for uric acid. So uric acid uh, is produced from inosine through this pathway here. And so in lower mammals like mice, for example, the uric acid levels are fairly low because there's an enzyme called uricase that degrades it essentially or metabolizes it through this pathway to urea. Okay, but the birds and bats have a mutation in this uricase enzyme, and so the uric acid accumulates at higher levels. Also, humans have a mutation in uricase, and that's why humans have relatively high uric acid levels compared to mice or rats or most other mammals. In fact, this is from a study done by Richard Cutler a long time ago. And what he did is he got blood essentially from a bunch of different species of primates, New World primates like us, Homo sapiens, chimpanzees, gorilla, orangutan, and then some older world monkeys, and measured uric acid levels and also vitamin C in some of them. But what he found here is that these longer lived species of primates have higher levels of uric acid, um, 5 to 10-fold higher, uh, or even more than these shorter lived primates. So that's a correlation. We got interested in this uric acid relationship with aging in the 1990s. And we actually asked the question, okay, mice and rats, they have low levels of uric acid. Also, it had been shown by actually, among others, Richard Cutler's son, Roy Cutler, who was a biologist in my lab, that uric acid is a really good antioxidant. It can sop up free radicals. In fact, it's maybe the major antioxidant in the blood. So we administered uric acid to rats, and then we subjected the rats to a model of stroke in which the middle cerebral artery that supplies blood to cerebral cortex and striatum, a brain region that controls body movement. Uh, and so when we shut off the blood supply over periods of hours and a few days, nerve cells die. And these images on the right show brain slices. The white is the damaged brain tissue area. So this is cerebral cortex here. This is striatum here. And so these animals have motor dysfunction, which we can measure, give them a neurological score. How good are they at maintaining their balance on a rotor rod or hanging on a string, essentially? And um, and so we had control animals that were just given placebo, and then animals that were given uric acid, essentially in a slurry because it's so insoluble. And what we found, and we measured the amount of brain damage. And so in the control, the placebo-treated animals, you get this much brain damage and this much neurological function, dysfunction. And in the uric acid treated animals, there's much less brain damage. You can see that in these examples and quantitatively here, and data from many animals, and um, and less uh, functional deficits. So uric acid is neuroprotective. Uh, More recently, now, uh, whatever, 30 years later, a three-center preclinical trial also found that uric acid administration is protective in stroke. And hopefully someone will do a clinical trial of this, just acutely giving uric acid after the stroke and with the idea that it'll protect their brain, improve the outcome. And then and the nice thing here, you can administer uric acid in principle into a blood vessel uh, that goes into the brain, so you don't even have to get that much uric acid circulating through the whole body. And then just kind of an interesting progression of the science here. Since uric acid is fairly insoluble in water and blood, we asked the question, can we maintain the antioxidant activity of uric acid while increasing its solubility? And so what and this involves a chemist, Nigel Gregg and Frank Haberman, and these are in my laboratory at the National Institute on Aging. And they took uric acid molecule and added methyl groups, CH3, or sulfur groups, S, to different places on the uric acid molecule, many focusing on these three different uh, carbons here. And then these were screened in various models to test their antioxidant activity. And then we tested them in the stroke model. And we found that several of the methyl group modified uric acid molecules were very effective in protecting the brain and reducing dysfunction, neurological dysfunction, brain damage here. And and so those are the kinds of molecules that maybe could be moved forward to the clinic to avoid any adverse effects of precipitation of uric acid. Uh, this is a study. Uh, there's this notion, and it's in the historical literature, I think maybe beginning in the Greeks or Romans or that time, that that individuals who eat a diet that leads to high uric acid levels and development of gout, so a lot of meat, for example, um, so the rich nobles, uh, leaders in those societies, that their brain function better than, and this is kind of a sort of a classist, racist type thing, but then others. So there's this idea that uric acid can affect cognition or intellectual activity or even mood. And so that this was thought of not by myself, but by a member of my lab, Simo Camandola, to, and then she collaborated with people working in the Baltimore longitudinal study of aging and another aging study in Sardinia, Italy. And what they found is a correlation between blood uric acid levels and impulsivity. Um, you know, making quick decisions without a lot of thought and consideration. So that was kind of interesting. And then also tested this uh, in mice and found that if we knock out the uric acid uricase gene in mice, okay, so that they have higher uric acid levels, then those mice have higher levels of kind of uh, exploratory behavior, less anxiety. So they a little more impulsive and less anxious. Um, More needs to be done looking at how uric acid affects the brain. Uh, that's pretty much it that's been done. Okay, so does uric acid explain increased longevity of bats and birds? They have high uric acid levels. They live longer. Uric acid is an antioxidant. It can protect the brain. Uh, Not much has been looked at beyond that. Here's another interesting creature here, the naked mole rat, which is a small rat, lives underground. As you can see, it's hairless. That's why it's called naked mole rat. But these rats can live more than 30 years. And moreover, whereas cancers is a major cause of death in rats and mice, these creatures apparently don't ever develop cancers. There's been none identified, and they die from other causes. Actually, the cause of death is not well established. Um, and there's a couple of things that have been reported, differences between these long-lived naked mole rats and rats and mice in the laboratory. One is that these naked mole rats have high levels of this molecule, hyaluronic acid, which actually people put this in creams on their skin because it seems to be beneficial for the skin, maybe counteract skin aging. And this molecule is normally present between cells in the extracellular matrix. And so there's some notion in relation to cancer that it might suppress cancer by somehow preventing the spread or the growth of the cancers. Um, but uh, since this since this paper was published here in Nature, there's been a lot of work showing differences in the immune systems of the naked mole rats and relating that to cancer. And these naked mole rats, their cells seem to be able to repair DNA damage better than their uh, short-lived counterparts. And also, if you look at the profile, and I put the references here if you're interested, the profile of the immune cells, there's changes that suggest that their immune system is better able to detect and eliminate precancerous or cells in the early stage of cancer formation before they're able to proliferate and become resistant to death. So that's interesting. And these kinds of studies, the Nick and mole rat, the uric acid story, show you the power of doing comparative um, interspecies comparisons. So I mentioned the aspen tree. So this is one tree here because this whole grove, if you will, started from one tree sending out rhizomes under the ground, and this is another tree. So this kind of like raspberry bushes, which many of you are more familiar with, where they send out these rhizomes. So you start with this, and then you end up with what looks like a lot of different plants, but it's actually all from the same plant through the underground uh, uh, propagation. Now, some species of trees, we actually don't know the longest lived trees. They may be immortal in a sense, propagating by these rhizomes, but at least 10,000 years for many of these. Okay, so in the last part of this lecture, I'm going to focus on animal models. What are the most commonly used animal models? And I'll present these animal models, and then in subsequent lectures, particularly the lecture after this, I'll talk about how these organisms have contributed to understanding aging in general and brain aging in particular. So yeast, well, yeast don't have brains, but some interesting genes were discovered that affect lifespan in yeast first, and then later shown to play a role in lifespan in insects and mice and maybe humans. And also in yeast, the process of autophagy was discovered, this mechanism whereby cells eliminate molecular garbage, very important in protecting against aging and disease, particularly neurodegenerative disorders, which I'll talk about in a lecture. Um, Worms, particularly a C. elegans, which every cell in its body and nervous system have been identified. There's a specific number of cells, and the developmental origin of all of those cells is known. So the parents of cell X is known all the way back to the first cell division. Um, Flies, Drosophila, um, again, powerful genetics. In both these worms and the flies, you can manipulate any gene you want up or down, uh, pretty much at will and increasingly very easy. So it's easy to do genetic manipulations. They have short lifespan, which is some advantage for aging because you get answers back quick. So these worms and flies typically live maybe 3 weeks or so. So you can do genetic or environmental manipulations and see what effect it has on lifespan very quickly. You can look at the effects on the aging nervous system. These organisms, uh, their nervous system function declines with aging, and that can be quantified in various ways, looking at movement of the animals or their ability to learn and remember things, simple learning and memory tests. Um, Mice and rats are very valuable because they're similar to humans in terms of their organ systems. They have essentially all the same organ systems, and development of those systems is pretty similar, although different rate of development. They develop many of the same diseases, cancers, uh, kidney disease, liver problems. They generally have less spontaneous development of neurological disorders, but certainly their neurological function declines with aging. Genes can be manipulated in mice. Their lifespan, 3 to 4 years, is long, so it takes a long time to get data back on lifespan. Also, they're expensive, relatively expensive to maintain. You need staff and make sure they're fed, watered, and their health is monitored all the time. And so that's expensive, particularly over 3 to 4 years, and even more resource intensive is non-human primates. And there's ethical concerns with all of these animals, right? Keeping them in a laboratory environment, not in their normal environment. Um, right, and this shows kind of the weight and life expectancy of these different species. Not much to say here beyond the previous slide, uh, but there's reasons these particular species were have been used so much. One is length of lifespan, about two is genetic manipulation and relative similarity or not to humans. But genetically, a lot of the genes are conserved pretty much from yeast on up to humans. Speaking of yeast, here's a yeast cell that's dividing. So as I mentioned, when these cells divide, you have a after the division, you end up with a small daughter cell and a larger cell. And this cell will generally survive, and this one will die. And as these are dividing, the damaged proteins, nucleic acids, membranes, organelles that have accumulated damage as the the yeast grows are segregated to the smaller cell, and then after division, this dies. So the mechanisms for this sorting have been worked out, at least to some extent. It's pretty interesting. Another interesting thing to mention here at this time is that kind of a similar process occurs in mitochondria, in cells in our body and brain. And as you likely know from an evolutionary perspective, mitochondria originated from bacteria developing a symbiotic relationship with another cell. And that symbiotic relationship gave that cell a survival advantage and has been maintained uh, in us and all these other organisms I've talked about. But point here is when mitochondria divide, similar to yeast, they segregate damaged dysfunctional parts to one daughter of the mitochondria, which then bud off and those dysfunctional damaged part of the mitochondria are moved to the lysosomes in the autophagy process. We call that mitophagy. So that's removing uh, parts of mitochondria that are damaged. And then you preserve functional mitochondria. This is just looking a little more detail on showing the kinds of things that cause molecular damage, reactive oxygen species, so free radicals, and the segregation of parts. And uh, yeah, so these are things like oxidatively damaged proteins, protein aggregates, and this becomes very important when I talk about neurodegenerative disorders like Alzheimer's, Parkinson's, ALS, frontotemporal dementia, all involve abnormal accumulation of protein aggregates in neurons and those aggregates are not removed by these kinds of processes. So there's a age-related inability of neurons to remove dysfunctional and aggregating proteins, and that can uh, enable the development of diseases. This is coelacanth, the roundworm that lives in the soil. This is the lifespan here and the developmental period. I should say that very interesting feature of these worms that I didn't mention yet is that if you crowd them, if you put them at a high density in their environment where there's a lot of competition with other individuals for a limited amount of food, which they eat mainly bacteria, and that's what's given to them in the lab. Or if you just, keep them at a low density and don't give them much food, uh, they go into what's called a dower larva. It's a dower larva state where they quit growing and they seem not to age in that state. So they're not eating, they're not growing, they're kind of dormant. Uh, these are kept mainly at room temperature, so they're relatively low temperature. And then if one puts food back in the cage, or not in the cage, in the dish where they are, they can come out of this dower larva state and continue development. So that's kind of interesting in relation to caloric restriction in mammals. Also, every neuron in this organism has an identity, uh, a name, if you will, and the connections of every neuron with other neurons is known. So the whole nervous system is mapped out. Uh, One can use genetic manipulations to knock out or 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 put in genes in any neuron or groups of neurons uh, one wants to, and see what effect that has on whatever you want to look at. And there have been studies, I won't talk about them, I guess, in which manipulation of, well, I will a little bit indirectly, manipulation of a single gene in only a few neurons can extend the lifespan in these worms. That's pretty interesting. This shows the so-called connectome of the worm. So each of these dots is a neuron, red and blue dots, and then there's lines between them showing how their axons, how they're interconnected with their neurites. Okay. Um, so some of these neurons, for example, control the pumping behavior of the animal, which essentially sucks in bacteria and takes it up and gets it into their intestines. And they have kind of this constant pumping rhythm. And during aging, that rhythm decreases and becomes irregular. And that's because there's problems in the neurons that control that rhythm. Now, one pathway that's been studied heavily in these worms, in fact, uh, uh, yeah, really, that was the animal model that really led to this hypothesis that the insulin/insulin-like growth factor signaling pathway may play an important role in aging. And the evidence was if you genetically knock out this insulin-like receptor in the worms, they live longer. If you knock out this downstream kinase, they live longer. And if you knock out this transcription factor activated through this pathway, uh, they live longer, and that's shown here. Okay, so that's interesting. That indicates that this cell growth pathway is actually accelerating aging. If you inhibit the cell growth pathway, the worms live longer. And however, in translating this to, say, mice, rats, and humans, it's not so clear what this means. Um, it could be consistent with the idea from that insulin-like growth factor signaling pathway because that's actually connected to the growth hormone IGF signaling pathway. It's probably less relevant or maybe even contradictory to the insulin signaling pathway itself, which, as you know, insulin resistance and diabetes essentially accelerate aging and the development of age-related cardiovascular disease and other diseases. Um, so that has to be worked out. But this kind of interesting. This pathway seems to be important in lifespan regulation. Here's just some examples knocking out the kinase downstream of the insulin-like growth factor pathway in the worms, and they live longer. And same with the DAF2, the receptor knockout, they live longer. And also, I mentioned you can look at certain behavioral changes with aging. These worms will move towards an odor placed in one edge of the dish, agar dish, essentially that they're living in. So the chemotax toward that food source, they'll smell it essentially and move towards it. That ability declines with aging. And another interesting thing is uh, in these worms in which lifespan is extended by genetic impairment of the insulin-like growth factor signaling pathway, um, these worms are more resistant to a variety of stresses. Shown here is heat exposure. They more survive longer when exposed to high heat, more survive longer when exposed to ultraviolet light, which causes DNA mutations. Um, and so these may have better DNA repair and better, for example, uh, production or function of heat shock proteins that are induced in response to heat. Um, so if we look at this in relation to caloric restriction, um, so caloric restriction extends lifespan. It does two things. It will reduce IGF uh, signaling and inhibit the mTOR pathway. This mTOR pathway, which I mentioned briefly previously, it's a pathway for protein synthesis. So um, when an individual eats, amino acids and glucose and fats increase in the blood. The mTOR pathway is activated, and that promotes cell growth, protein synthesis, which is required for the growth of cell. However, activation of this mTOR pathway also inhibits autophagy. So with caloric restriction, then cells essentially slow down or quit growing, and they increase their autophagy, and that's associated with increased longevity. And, and the opposite way, if you overfeed animals, particularly with high protein and high fat, actually, at least in animals, high glucose, high protein, autophagy decreases and the animals don't live as long. Uh, Third model, Drosophila, third model. This is the lifespan of these flies. Um, their brain as well mapped out, the connectome of the brain, if you will. Individual neurons can be visualized by putting fluorescent probe in them through genetic means. Genes can be manipulated and studied. And as in the worms, manipulations of the mTOR and insulin-like growth factor signaling pathways can affect lifespan, as well as energy restriction. So this just shows us in more detail. Inhibit the mTOR pathway, longer life. And later I'll talk, I think, about you can just restrict the amount of protein you give to these animals, particularly certain amino acids, and extend their lifespan. Also, pharmacological, that is, drug-based inhibition, selective inhibition of the mTOR pathway extends lifespan. Um, yeah, so I mentioned this leak. So growth hormone is a hormone produced by your pituitary gland, and it's high levels in humans during early life and promotes growth, bone formation, uh, muscle formation, and it will induce insulin-like growth factor production mainly from kidney cells. But this IGF-1 is also produced by brain cells. So that promotes this rapid growth and elegant construction of the body and brain during development. Um, Dietary restriction will inhibit this, so you don't want to inhibit energy and protein availability, certainly during fetal life or early postnatal life. But once you reach sexual maturity, then um, activating this pathway may be detrimental. Um, this more detail on this. I will go into a little more detail on the mTOR pathway later, but the pathway is well worked out and um, and effects of manipulations studied. I should point out that this mTOR pathway is important for growth and also mitochondrial biogenesis, the growth and division of mitochondria. This just shows that rapamycin, that chemical that inhibits the mTOR pathway, was actually discovered uh, based on some colonialist colonizers going to Easter Island and taking soil sample back to England and isolating this chemical from bacteria and then showing that this chemical can actually inhibit the growth of certain microorganisms, but then later showing that it can inhibit the mTOR pathway selectively. And then this is from a three-center study of looking at the effects of rapamycin on lifespan in mice. So there's about a 10%, actually up to a 10% increase in average lifespan of the mice with rapamycin, no change in maximum lifespan. So this is studied from hundreds of mice. One problem with these studies is that rapamycin inhibits the immune system. In fact, it's used to treat patients that are getting organ transplants, and these mice in this study and most studies, 99% of all studies, these animals are maintained in rooms that are pathogen free. They don't have any infectious agents present. So their immune system is unchallenged throughout their life. And and so the point there being that in humans, taking rapamycin long term may not be a good thing if it's inhibiting immune function. It may increase your susceptibility to COVID or whatever, infectious agent, bacterial infection, etc., pneumonia. But this is proof of principle that inhibiting the mTOR pathway can extend lifespan. From the standpoint of safety, it's better to inhibit the mTOR pathway through dietary energy restriction, intermittent fasting, or perhaps protein restriction. Okay, so that's the first in this five-part lecture series. Now, I'll quit there, and I hope you enjoyed that, and a lot of information there, food for thought. Had references to pretty much everything I talked about there. If you're interested in getting those references, you can probably find most of them on PubMed. If you can't, you're welcome to email me. I can get them and send you the PDF. Next time I will talk about kind of three different things focused on the brain. So the rest of this is going to be totally the brain. Um, I'm going to talk about, um, essentially, I'm going to talk about functional changes that occur during normal brain aging, structural changes that occur during normal brain aging, change in synaptic connections, number of neurons, neurogenesis, etc. And then I'm going to talk about uh, biomarkers. Uh, I'm going to talk about biomarkers of brain aging mainly, actually in the next talk, and then later talk about the functional and structural aspects, and then talk about mechanisms of neurodegenerative disorders, approaches to preventing or treating these neurodegenerative disorders. And that will be the 5-part series. So thanks for your attention. Bye.